Hey guys, I just made a whole video that did not record, so we're gonna do a slightly modified version on my phone that I trust a little more than the document camera I brought home. So, I'm gonna try to just hold this here. So, crossing over is this event that happens in prophase 1 where genes can kind of get swapped. So if we think about right here, so maybe this is a chromosome you got from your mom, and this is a chromosome you got from your dad. They both have been copied already. What happens in prophase 1 is they get right together like this, and you see they're kind of hugging. Okay, they call this, they embrace, and some of their genes actually trade places. So this is the result at the end of prophase 1 where they have had crossing over. So this dominant C is now on the chromosome from dad, and this recessive C is now on this chromatid from mom. So they trade places here. Okay. So this group of 4 chromosomes is called a tetrad. And so this structure right here is what we would call the tetrad. Okay, so there we go. Forms during prophase 1 and is the structure that is the 4 chromatids, 2 from the chromosome from mom, 2 from the chromosome from dad. So when we're looking at meiosis, okay, so the top row is meiosis 1 and the bottom row is meiosis 2. Okay, and if you notice in meiosis 1, they're lined up 2 by 2. So there's 2 chromosomes there, 1 chromosome plus its homologous partner, the one from the other parent that has the same genes. Okay, and so they're 2 by 2. Homologous partners together, homologous partners together. Where in meiosis 2, they're lined up single file, 1 chromosome, 1 chromosome, 1 chromosome, and they get pulled apart. So meiosis 2 looks exactly, excuse me, there it is, like mitosis, whereas meiosis 1 has differences because of those homologous partners. So in anaphase, each chromosome has a 50% chance of going each way. So again, let's kind of think about that. Okay, so here the red one goes up and the blue one goes down, but it could have equally gone the other way. Okay, this one, same idea. The red one went up, the blue one went down, but it could have just as easily gone the other way. It's kind of random. And so we take this random 50% chance times all the 2 to the 23rd, because there's 23 chromosomes. And so we end up with that many possible combinations just for one parent. Okay, so if mom has this many possible combinations and dad has this many possible combinations, you can only imagine how many possible different children 2 parents could have. And this is not even counting the crossing over, the variety we get from this process up here. So in reality, there's a whole bunch more than even that many. Okay, last page. So problems with meiosis. Okay, so sometimes those chromatids that are supposed to get pulled apart, they get stuck together. And that is called non-disjunction, and it happens during anaphase 1. And if you think about this, if there's a problem with a gamete, okay, and then the gamete gets with another gamete, forms a zygote. Zygote has all of the chromosomes of the new organism. When that zygote does mitosis, it's going to pass that deformity on to all of its cells. Okay, so you could test any single cell of an organism that's a result of non-disjunction, and they're all going to have this deformity. So most of the time, they're called trisomies. They're going to have an extra copy of a chromosome. And the most common one is called trisomy 21, where they have an extra 21st chromosome. So if you think about this, here was our picture of all the normal chromosomes. Trisomy 21 would look like this, right? So the 21st chromosome, there's kind of an extra one there. Okay, so one of the gametes, instead of bringing one of the 21st chromosome, had 2. So they have extra information. So they're going to make extra proteins, and it's just kind of mess up the balance of how things get done in the body. And so trisomy 21 is the more technical name for Down syndrome. Alternatively, you can have an organism with just one chromosome instead of two, and that's called a monosomy, which is usually fatal, meaning those organisms don't live. But one example of one that's not generally fatal is called Turner syndrome, where instead of having an X and an X, like most girls are XX and boys are generally XY, Turner syndrome, there's just one X. Okay, so of the sex chromosomes, they just have one. So they don't have the information on the Y chromosome to make them a boy, but they also are going to have less copies of all the genes that make them female. So these are generally going to be short females that are without scientific intervention sterile, meaning they can't have their own children, but they can still have, like I said, lead very normal independent lives. And then we have this final idea of the chapter called gene linkage. So our law of independent assortment from earlier said that the likelihood of one gene being passed does not affect another gene. Well, that's not always exactly true. If two genes are on the same chromosome, then they can affect each other. Okay, so I drew kind of down here. Here's a chromosome with 6 genes on it, A, B, C, D, E, F. Here's a different chromosome with 6 different genes, G, H, I, J, K, L on it. And the likelihood of getting this one or this one are random, not affected. They would follow the law of independent assortment. But if two genes are right next to each other, they're neighbors, unless crossing over separates them, they're going to be passed together. Okay, so the closer together genes are on a chromosome, the more linked they are. Okay, they have to be on the same chromosome to be linked at all. And if they are linked, the closer together they are, the more likely they are to be passed together. All right, that's it for chapter 10. If you have any questions, shoot me a message on email or Google Hangouts. I'm working to try to get a Zoom figured out, hopefully for Tuesday during our normal 3rd and 5th period times. We'll see if that works out. Pay attention on Google Classroom. Please, please. All right, bye.